Okay, guys, it's 12 o'clock, and welcome to Cancer Center Grand Rounds. A couple of announcements. Just in case anyone didn't know, next Wednesday, the Cancer Center grant will be reviewed by 28 of our closest friends from around the country who will be joining us for the site visit. We'll be having our site visit up at Bet's house in the Greenberg Center. And I want to thank all the members of the Cancer Center who have worked so hard to put together what I think is really a terrific presentation and something which I hope will be received warmly by the site visitors. So thank you to everyone for that. The second thing to announce is next week's Grand Rounds, we have a special visitor who's coming all the way from University College London. Dr. Chris Boshoff will be giving Grand Rounds next week. Chris is the Cancer Center Director of University College London. He works at a number of different areas, including DNA repair, also including the area of bladder cancer. He directs a diverse research group at UCL and has been a terrific partner with the Yale-UCL relationship. And he'll be here as well to participate in the site visit. We've asked him to give Grand Rounds, so we look forward to hearing from Chris on Tuesday of next week. So we now, this week, have our regular format of Grand Rounds, and we have two spectacular speakers. The second speaker will be James Yu. James is from the Department of Therapeutic Radiology here at Yale, and he'll be talking to us about prostate cancer and bending of the cost curve. Our first speaker is Dr. Donald Engelman, who is the Higgins Professor of MB&B. Dr. Engelman, I was just saying to him, Dr. Engelman was an established senior professor when I was a college student. And then Dr. DeMeo came up and he said, Tom, Dr. Engelman was an established senior professor when I was a college student too. So Dr. Engelman's been an established senior professor of MB&B for a long time, and we're delighted to have him today talking to us about tumor targeting and drug delivery using the PHLIP peptide. Dr. Engelman.
Thanks, Tom. Am I live? Yeah. So it's been a really wonderful experience becoming engaged with the Cancer Center and forming some collaborative relationships, and most of them I won't have time to talk about. My aim today is to tell you about a peptide that we discovered by accident. I'll tell you a little about the accident, but it is kind of a story that you might think of as a model for why the NIH invests in basic science, or at least if it actually turns out to be practically useful, you might think that. So I'm a biophysicist and have been, as you just heard, for a while. And so it was really quite a surprise to me after working on abstract issues having to do with molecular properties that I found myself working on these. I've learned this is a mouse, and I know a lot about them now. That's the front end, that's the back end. And this mouse has a tumor in it, and when we inject the peptide I'll tell you about, either IV or IP, it goes around and targets the tumor. So here's the peptide in its fluorescent labeled form. And so that's pretty much the message. You can leave now, or you can stay and I'll tell you some more details. So the kind of operative question that we've come to bear down on is whether acidity can be a useful biomarker for tumors. As we know, many biomarkers have been used to target tumors, and the general experience is that, here's the case of Herceptin, the general experience is that they do produce a benefit, but the benefit is modest. Viewed in terms of long-term survival. So compared with chemotherapy alone, Herceptin increases average survival by about 5 months. Well, that's valuable, but what one hopes for is more. But then we might ask, well, why is it that this curve goes down that way? What is it about tumors that they such that they become resistant to something that, in this case, binds to a surface receptor. And the answer, at least part of the answer, appears to be that real tumors are quite heterogeneous, and they express whatever the marker is at varying levels in different cells in the tumor. And so you apply whatever the targeting molecule and therapy is of your choice, and it gets all the cells that express a suitable level of that marker, but then the rest of the cells continue to grow, and the tumor eventually recurs, and with the result that the curve goes down. So the question on the table is whether acidity might be something that could be targeted. Tumors are acidic, and they're acidic for a basic reason having to do, or a set of basic reasons, having to do with their physiology. So this is a marker that can't be selected against except by not being, by converting to a non-tumor phenotype. So selection seems unlikely. And the peptide we've discovered is a way to target acidity. So the reason for being interested in this is, I think, that it's a different route to target tumors based essentially on their physiology. What is that physiology? Well, one basic property is that tumors are often growing too fast for their oxygen supply, or trying to. And so the result is that the lack of oxygen puts them into anaerobic respiration. They use the glycolytic pathway that you all know very well, and yes, I could examine you on it, but I won't. And the cytoplasm becomes acidic, and the cells pump the acid out to maintain their physiological balance. That means that the environment gets acidified. The other, another major contributor is that cancer cells take up glucose very avidly, as probably most of you know. And the result is that they tend to overwhelm the capacity of their mitochondria. And the result is that even in the presence of oxygen, the glycolytic pathway gets used a lot, acidity is produced. And furthermore, many tumor cells express carbonic anhydrases at their surface to deal with the carbon dioxide that's produced by metabolism. And that too acidifies the local environment. So there are several contributors to acidification of the extracellular environment in a tumor. Now, we were studying protein folding, and we had the idea that you could think of the early stages of membrane protein folding in terms of the formation of independently stable helices and then their side-to-side interaction to initiate the development of the folded state. And this turns out to still have some merit as a thought about how things emerge from the translocon, and we could have a whole other lecture here, but we won't. But a part of this idea involved the proposition that the individual helices in a polytopic membrane protein would be independently stable. And there are a bunch of arguments connected with that. But it set us off to test this idea. John Hunt, when he was in the lab, did it by taking the protein bacteria rhodopsin, which has 7 transmembrane helices, and he took each one, put it in a peptide, mixed it with lipid in a suitable way, did a reconstitution, and indeed most of them then formed an independently stable transmembrane helix. However, the third helix, which was then called C, you do the same experiment and the peptide was found largely in solution. This was a very surprising finding for a transmembrane helix. But when we looked at the sequence, we realized that there are some carboxyl groups that are part of the transmembrane region, so we lowered the pH. And when we did that, the peptide inserted across the membrane to form a transmembrane helix. So this was an interesting result that we published a while ago, and it wasn't until Jana Reshetniak and her husband Oleg Andreev joined the lab in different roles that we became aware that this might have applications in thinking about tumor targeting. We now call these peptides PHLIPs. This stands for pH low insertion peptide. I know, but we get to play. And so here's the sequence. This is the part from bacteria rhodopsin. If we zoom in on it, we see that there are 2 aspartic acid groups and a somewhat buried tryptophan. This is very useful for following the behavior of the peptide, and we used it to investigate the process of insertion. Because it's a peptide that we synthesize, we can put groups like cysteines in different positions to use chemistry of different kinds to attach ligands. Lots of biophysics later. We know that several things about it. We know that it is soluble in as a monomer at low concentrations in aqueous environments, and that it's unstructured. If there's a membrane or a lipid bilayer present, either a synthetic lipid bilayer or a biological membrane or cell, the peptide binds pretty strongly to the surface, still in an unstructured form. And if we lower the pH, it inserts across the membrane to form a transmembrane helix. And we can follow this by fluorescence. Here we see it in solution on the membrane and inserted, or we can follow it by circular dichroism, which is a measure of secondary structure. Here we see it unstructured in solution, unstructured on the surface, and inserted to form the transmembrane helix. This spectrum is oriented circular dichroism from a stack of lipid bilayers that tells us that the orientation of this helix is in the direction perpendicular to the bilayer. So some of the properties, I mentioned it's water-soluble, forms a helix. I didn't mention that it's the carboxy terminus that inserts across the membrane. And so it's directional insertion. It's monomeric to the extent that we can measure it at low concentration in all of these states. And that's really quite remarkable. And it leads another branch of the work is to use this as a model to study how peptides insert into membranes, because it's extraordinary that you have a peptide that's soluble in water and that can form a transmembrane helix. Both of those things are usually incompatible properties. Doesn't seem to harm cells at all. We've done a bunch of studies to look for membrane fusion events or leakage events, and when injected into mice or put into culture with cells, there doesn't seem to be any toxicity that we've yet measured. We haven't done a formal tox study yet. And from the standpoint of biological, its use as an agent, the fact that the D-amino acid peptides work as well as the L-amino acid peptides means that it's a couple of things. One is it means that it can't be receptor-driven or have anything to do with specific interactions with anything in the cell, because the D-amino acid peptide shouldn't do that, but the L-amino acid peptide does. And it's also more stable, one thinks, in circulation. So I'm going to say a few words now about how it works, what the mechanism is. By doing kinetic studies, we were able to show that the helix forms fast. It's a very difficult experiment to do rapid kinetic studies using circular dichroism. It's very easy using fluorescence. So we follow it both by the CD signal and the fluorescence signal. These lines are actually fits of an explicit model. They aren't just multiple exponential fits. And we get time constants. And notice that this is really pretty slow. So this is on the seconds, many tens of seconds time scale for the insertion event. But the helix formation goes fast. So it forms a helix on the surface and then inserts. If we go the other way by jumping, this was for jumping the pH from 8 to 4, so from toward acidity. If we jump away from acidity, then the helix unfolds and emerges pretty much with similar time constants. So it's a different pathway, and we've developed some molecular fantasies about what might be going on, and we're trying to test them. But basically the idea is, once again, you start here, you form a helix, and then somehow it winds up slowly as a transmembrane helix. We've also done a lot of what's been called protein abuse. We've done a lot of alterations of the sequence. I'm just going to focus on these two positions. These are the two carboxyl groups that we think are most principally involved in the insertion event. And when we change them to K, that's lysine, the K flip with two lysines doesn't insert. If we change them to asparagine, once again, doesn't insert. If we change them to alanine, we never get to know because the peptide aggregates for some reason that we still don't particularly understand. Except that maybe the charge or the polarity of the aspartic acids helps to keep it soluble. So we think that the mechanism involves the protonation of carboxyl groups, and that triggers insertion by rendering those groups more hydrophobic once the charge is removed by adding a proton. And interestingly, when we substituted with glutamic acid, it worked fine, but it changed the pK of insertion. So that and subsequent experiments have shown that we can tune the peptide to different acidity levels, and that could be clinically important for targeting tumors successfully. And here's an experiment in which the different variants were tried, the one with lysines and the one with asparagines, shown here and here, with Psi 5.5, a dye that emits in the infrared fluorescence, has infrared fluorescence, targets the tumors if it's FLIP, but does not target them if we change these groups. So it's nothing about the binding of the peptide or something like that, we think. So what we know that when the pH is low, we form a helix and we get insertion. The aspartic acid groups are a key for the insertion event. Changing them can abolish it or alter the insertion pK, and other groups modulate cooperativity. Not discussed. So we know how to manipulate some of the properties of this peptide with respect to insertion across membranes, and then the question for this group and for us is, how can we use these molecules for diagnosis or therapy? So we have three lines of work. One is imaging. We attach something, remember it was unidirectional insertion. So we attach something on the end that doesn't go through, and we use the partitioning energy that at low pH of putting this across the membrane to locate that entity at the membrane in an acidic environment. The second idea is that we attach a molecule to the inserting end using a disulfide. And the disulfide, if this is a cell, once it gets inside the cell, the disulfide gets reduced because there's the reducing environment in the cytoplasm, and the molecule, whatever it is released. So that's a delivery strategy. And then finally, something I'm not going to speak of today except for this, is the idea of using this phenomenon to target either liposomes or nanoparticles to cells in tumors. And we're beginning to work in that direction with our collaborators, and there are some promising observations there. So first, what about targeting tumors? So I've already shown you a couple of images. I'll show you one more here. So this is a study with putting in cultivating cells that express GFP. Doesn't show up so well because the GFP fluorescence doesn't penetrate tissue very well. And here are the images using fluorescently labeled PHLIP using an infrared dye. Actually, lately we've been preferring Alexa 750. The infrared penetrates tissue well, and so we can image it better in a whole mouse. And so this is the same mouse progressively. And if we look at the contrast index, we see that it increases nicely. If we use this K-PHLIP that I mentioned before as a control that doesn't insert, nothing happens. And if we add glucose just at the time of administration, just one shot, it enhances the uptake and development of the contrast index very nicely. We've begun in collaboration with Jason Lewis's lab some experiments to look at imaging using radioisotopes, and we'll probably do some work in that direction here as well. And here are a couple cell lines. This is a more aggressive cell line, and you can see that the tumor imaging, these are contrast indices here and here. Tumor imaging is better. Here's PC3 cells, and you can see still get some imaging. Tumor targeting correlates with tumor aggressiveness. So if you've got a tumor that is faster growing and more aggressive, then all those metabolic signatures that we talked about will operate more forcefully. The Warburg effect, the Pasteur effect, those things will be enhanced in tumors that are made of cells that are growing more rapidly, more aggressively. And indeed, if we look at M4A4 cells, which are a metastatic line, the labeling is much better than NM2C5 cells, which are not. And here again is the contrast indices. So interestingly, we may be able to distinguish to some extent the physiological state and degree of aggressiveness of a tumor using some imaging strategy. Model tumors, as I'm sure everyone in this room knows better than I do, in which you basically culture cells under the skin of a mouse, are not representative of a lot of aspects of real tumors, particularly cell heterogeneity and so on. But there are mouse lines. One of them is the tramp mouse, which is a mouse that gets a spontaneous prostate tumor. And here's a tramp mouse labeled now by IP, injection of PHLIP, and you have to wait for the background to subside, and then open the mouse, and there is the tumor. This mouse conveniently fell apart into all of its pieces, and when we looked at them, we could see that there's significant accumulation in the kidneys, which is not surprising because they're acidic. Here's the tumor. When you take the tumor and cut it in half, you see that the labeling is throughout the tumor. And this is actually quite exceptional, because many labeling strategies you'll see a halo here because the label won't get into the tumor. So somehow, and we don't know exactly how this works, the PHLIP exchanges its way into the body of the tumor. We've tried a couple of models of metastases, one by implanting a metastatic line, letting it go metastatic, removing the primary tumor and then looking, and another by injecting cells into the blood of a mouse. And indeed, both of them label well with PHLIP, right down to the cellular level. And once again, we're using a line that has GFP expression, and so when you look at the cell level, you can see that the PHLIP is kind of around the periphery of the cells and the GFP is in the cytoplasm, just as we would expect, given the labeling strategy. And this dot is a metastasis that's smaller than 2 millimeters, and so what we find is that we can successfully target things as small as 2 millimeters or less. Once again, comparing GFP, this is now opening a mouse with a tumor. We compare the boundaries indicated by the fluorescence of GFP with that from PHLIP. They coincide. If we take out the tumor and leave little bits behind and zoom in on them, the images still coincide, and this is a millimeter size scale, so these are sub-millimeter little bits of tissue that can be successfully identified, potentially opening the way to image-guided surgery. So tumors are targeted. There is a background in kidneys that we have to think about. Aggressive tumors are better targeted. Small tumors can be targeted. Now, I'm pretty much out of time, but I just want to say that we've done some studies with model compounds to establish what we can deliver into a mouse, and we have an attachment point. We vary the properties, and we detect by fluorescence. And here's delivery of 2 of those compounds into cells. And the properties of those molecules are surprising. That is, they are quite polar. So a negative log P is a polar molecule. A negative log P of 2 is really quite polar, and these molecules would not get in on their own, leading us to think that we could deliver functional molecules like peptide nucleic acids. So here's delivery of a peptide nucleic acid in the same kind of experiment, putting a fluorescent label on the peptide nucleic acid, delivering it into a cell, zooming in, and you see it in the nucleus. And our colleagues, and we're doing some parallel work here, our colleagues have done one experiment that I'll show you, although it's preliminary, with Peter Nielsen. And this is a tumor grown from cells that have a splicing defect that can be corrected by a peptide nucleic acid, and it appears that it is when you inject PHLIP carrying a peptide nucleic acid. So PHLIP can target and deliver molecules with no formal charges are a problem. Log P's quite polar, big into a cell. So the overall conclusions are we can target acidic tissues, including tumors. Cargo molecules can be delivered. Nanoparticles can be targeted. We didn't talk about that, but we've shown that nanogold can be targeted to tumors in the same way. So we have surface targeting for imaging. We have the potential of delivery into the cytoplasm, and we have nanoparticle targeting. This work has involved a lot of people, and on an ongoing basis it will continue to do so. I just want to pick out that Oleg Andreev and Yana Reshetniak are my close collaborators. We've been lately beginning some very important collaborations here at Yale with the Bosenberg Lab, the Glaser Lab, the Saltzman Lab, and the SLAC Lab. And we thank the YCC for facilitating this, and we've had a pilot grant, and we're rolling. So I'm very pleased to tell you this story, and I'm now open to questions. question. It seems to work either way. IP is a slower delivery. Takes longer to find its way into the blood. We believe that the reason PHLIP can stay around as long as it does is that it rides on the surfaces of the red blood cells. So the reason, if you inject a polar peptide into a mouse, it's usually gone in 10 minutes or less. Sorry? For metastasized. It might be that IP injection is better because it's essentially a sustained release strategy of a kind. I'm not sure I'm having trouble understanding what the question is. Oh, if we were going to use it, which would be the better route of administration? I'd say we don't know at this point. Yeah.
Can you kill or can you target single cells? Is there a sufficient pH environment around a single tumor cell? For instance, metastasis presumably originates from a single cell, probably from a single cell. Is there, I imagine that the pH is produced in a neighborhood of number of cells, but if you run a single cell, can you target that?
It's a very interesting question to which we don't yet know the answer. Can we target single cells? If you think about the pH at the surface of a cell, it depends in large measure on the delta pH across the membrane, which is part of the membrane potential. And because of the electrostatic potential, the delta pH will concentrate protons near the surface. So the pH at the surface, where this peptide is binding, is actually lower probably than the surrounding pH. So when you measure the external pH by NMR or by probe method or whatever method you do, the acidity is not really the characteristic of the cell surface. Cell surfaces are more acidic than that, particularly cancer cells. So it's plausible that single cell targeting might work.
Easy experiment to do.
Very good. We're going to have to move on. Let's thank Dr. again.